Once again, welcome to our science lesson. Remember, in our previous lesson, we were looking at how responses happen, and we said that responses happen due to existence of structures in our bodies called sensory organs. Sensory organs, we say they are made up of sensory cells. So if we have so many or a group of sensory cells being associated together or grouped together, and specialized in a way to form sensory organs. So one of the sensory organ is the eye. We have looked at several parts. Remember our diagram? We had mentioned parts like the cornea. We said cornea is a transparent layer that allows light to get into the eye. We also looked at the conjunctiva. We said conjunctiva is a thin, transparent membrane, usually lining the eyelid and slightly part of the eyeball for purpose of protection. So we were to look at other structures. We looked at the iris. The third part is the iris. Iris is made up of two muscles. Which are radial and circular. Muscles. But in our diagram, we were looking at iris from two-dimension point. So we were able to mention this as the iris. If that is the cornea, that is the aqueous humor. So these structures just after the lens or before the lens is the iris. This is a cross section or a longitudinal section, but if we do a cross section of the eyeball, this is how the iris appears. It's made up of muscles, two types. Radial muscles and circular muscles. So these two muscles, if you look at it directly in the eye of a person, you realize that this part is the one that forms the dark region, but it leaves a pore or a hole. That hole is called the pupil. But we have the muscles. This is radial and circular. So these two muscles are the one that forms that dark region, some colored region. It could be dark or any other color, sometimes not very dark or gray. So the major role of the iris is to regulate amount of light that gets into the eye. Regulates amount of light that gets into the eye. So the question is, how does that happen? We have the hole, the pupil, but now we have the muscles of the iris that will be involved. Let us look at this situation. If it is, for instance, high light intensity, During high light intensity, if this is the way we have just drawn, when there is high light intensity reaching your eyes, the two muscles are going to be involved. We have radial and circular. So for radial, it's going to relax. Radial muscles relaxes, while circular muscles contracts. Circular muscles contracts. So what will be the effect on the pupil? The size of the pupil, decreases. The size of the pupil decreases because when it contracts, it becomes even more smaller. So the pupil size decreases, becomes small. And so if there is high light intensity, it's going to reduce the amount of light reaching the retina. If it is high light intensity, high or low, that is low light, the opposite will happen. Radial muscles are going to contract. Radial muscles contract, then circular muscles relaxes. These muscles relaxes. They go back to the original position. When it goes back to the original position, the size of the pupil is going to increase. When it increases, it means it's becoming large in size. Therefore, amount of light that will be getting into the eye will slightly increase because the eye was training to get more light. So in these two ways, we can easily explain how the iris is going to regulate amount of light that enters the eye. We have said when there is high light intensity, the radial muscles are going to relax, circular muscles contract, and the effect is the pupil decreasing in size. So if it decreases in size, less light will pass through the pupil to reach the retina. And the opposite will happen when in low light intensity. The radial muscles contract, circular muscles relaxes, pupil increase in size, allowing more light to reach the retina. So in the process, it has controlled or regulated the amount of light that is that's getting through the iris to the retina. That's why when you look at a very bright light, the sequence of events that happens in the eyes are as follows: radial muscles relaxing, circular muscles contracting, decrease in pupil. That's why sometimes you will squint your eyes just to help the radial muscles contract so that they reduce the size of the pupil to allow in less light. Remember, we have said a lot of light reaching the retina usually damages the retina. We are going to find out how that one happens. The other part is pupil. We have already encountered the pupil in the iris. So that hole in the iris, the role is to allow light to pass through to reach the retina. Allows light to pass to reach or to pass to the retina. The other part is the aqueous humor. Aqueous humor. Remember when we drew the structure of the eye, we mentioned aqueous humor. So if, just a reminder, we said if that's the cornea, the curved area, we shall have the iris at that point, like that. But now we have those suspensory ligaments and the lens. So that area or that space found between the cornea and the lens, we gave it a name. We said it's called anterior chamber. Anterior chamber. Anterior chamber, we said it contains this fluid, the aqueous humor. So in here we have aqueous humor. Some of the roles of aqueous humor include roles of aqueous humor. One of them, this aqueous humor contains, it has nutrients and oxygen. So because of presence of nutrients, it provides the role of nourishing, provides nourishment to the cornea and the lens. Remember, it's the one that's in contact with cornea and the lens, and is having nutrients and oxygen. So because of that, it's supplying nutrients to these two structures, the cornea and the lens. And also because most of the material that's found in aqueous humor is water. And if water is contained in a very tight structure or container, it gives it shape. It gives it some kind of skeleton called hydroskeleton. So also gives shape, gives spherical shape, contributes to spherical shape of the eyeball. Indeed, that's why it's called a ball, because of the two humors, the aqueous humor and the vitreous humor. And then, just like any other liquid, because we have said most of the material that makes up aqueous humor is water. That's why it's giving it shape. And because of that, also, when light passes through water, there's some refraction. And aqueous humor having water, it also contributes to refraction of light until that light reaches the retina. So contributes to refraction of light for focusing onto the retina. Refraction is that ability of a liquid to bend light. So if, for example, that is the humor there, and we have an object at that point, light is coming to the eye, it will have to be refracted so that it can fall on the retina. Otherwise, most of the light will fall on some places where we do not have a receptor cell. So it is refracted. That light is refracted at that point. And then this light refracted at that point. So the image that is formed at the fovea centralis is real but inverted, although it could be small in size. So aqueous humor is also contributing to refraction. First part of refraction is the cornea. Second part is the aqueous humor. So the 3 points must be very clear: providing nourishment to cornea and lens, giving spherical shape to the eyeball or contributes to the spherical shape of the eyeball, refraction of light for focusing onto the retina. part number 6 is the lens. The lens. Part number 6, lens. Remember how the lens appeared in our diagram? They are held in position by suspensory ligaments. But in between this lens, we have a clear liquid, a clear jelly-like substance. A clear jelly-like substance. And then you look at the surface of the lens. It is curved but outward. When we have a lens curved outward, we call it convex. But it is made up of two sides. One side, the second side, two sides. So because of that, a lens is described to be biconvex in nature. So because of this, it is described as a biconvex structure. Biconvex structure with clear jelly-like substance. Clear jelly-like substance. Without forgetting to mention that it is held in position. It is held in position by this structure, suspensory ligaments. Held in position by suspensory ligaments. The role of lens. So majorly, the role of lens is to refract light. It's transparent to allow light, but the major one is to refract light so that it can be focused onto the retina. Refracts light to be focused onto the retina. To be focused onto the retina. That one comes about due to other factors like the suspensory ligaments. When suspensory ligaments, for instance, when the tension on suspensory ligament relaxes, when it relaxes, if it is relaxed, if it is relaxed, when it relaxes, the effect on the curvature increases. Curvature increases. When it increases, the focal length, that distance from here to there is called the focal length. The focal length increases. Curvature has increased. Focal length increases. There will be more refraction. So, more refraction. When there is more refraction, more light is bent to reach the retina. And the opposite happens. When suspensory ligament contracts, when it contracts, it pulls this lens. When it pulls the lens, the curvature decreases. So it becomes more flat. When it becomes more flat, there will be less refraction. When there's less refraction, if, for example, you are focusing for a distant object, an object that is on the horizon, that's what happens. So the suspensory ligaments will contract this lens lengthens, and in the process, the curvature decreases and there will be less refraction. Light falls onto the retina. So I've just been trying to explain how that refraction takes place. It takes place due to help of the suspensory ligaments, either relaxing or contracting. When it relaxes, this lens becomes, or the curvature of the lens increases. When it increases, more light is refracted. And then when suspensory ligaments contract, when they contract, they pull sideways. The lens becomes more flatter. When it becomes more flatter, there will be less refraction. When there's less refraction, if you are focusing for a distant object, the light now falls on the retina. Number 7 is suspensory ligament. Number 7, suspensory ligament. So we have already given some roles of suspensory ligament. We have said it's the one that holds. It holds lens in position. But that's not the only role. We have also said that it helps in focusing. In focusing for objects at different distances. We can go through it again. How does this one happen? We have said when, for example, let me use a diagram. We have an object that's very close to the eye, an object that is just close to the eye. Light will reach the cornea, but now we have suspensory ligament holding the lens. Suspensory ligament. So what will happen? Light will pass slightly refracted to the lens. What will happen if it is a close object? For a close object, if you bring an object very close to your eyes, sometimes the eyes even strain to look at it because there's a lot of tension loose happening. So what will happen? The suspensory ligament relaxes. When it relaxes, the curvature of the lens increases. It has increased at that point. When the curvature increases, more refraction happens. If that's the fovea centralis, more refraction is going to happen. And in the process, there's great bending of light. And our image will be formed at that point. It's usually smaller, but in an inverted position. So suspensory ligament have been involved, relaxing, increasing the curvature of the lens, bring about more refraction, hence a lot of light being bent so that it falls onto the fovea centralis, suspensory ligaments. And the opposite happens if it is a distant object. A distant object looks smaller, so by the time light reaches the eye, there's already a problem. So if that is the fovea centralis, where we have high concentration of cells to receive light, what will happen? The opposite will happen. There is contraction of suspensory ligaments. When they contract, they pull the lens. When they pull the lens, it becomes more flat. not as flat, but slightly flat. In other words, the curvature decreases. From our previous example, we said the curvature has increased, but right now it has decreased. So when the curvature decreases, there is less refraction. So less light will be, there will be less bending of light as previously. And an image will be formed at that point. Image is always inverted. So again, for a distant object, suspensory ligaments contract, the lens are going to become more flat, or the curvature decreases, and in the process less light is refracted and the image falls on the fovea centralis at that point. So this process of focusing for different objects at different distances is called accommodation. Focusing for objects at different distances is called accommodation. Structure number 8, ciliary body. Ciliary body or muscles. Ciliary body. So this one is also made up of two types of muscles. Ciliary body is made up of two muscles, smooth muscles and circular muscles. Remember, when we drew the eye, we have our ciliary body there, and onto it we have the suspensory ligaments. So the role of ciliary muscles is also involved in the same process as that one of suspensory ligament. It will contract and relax. Contracts and relaxes. When it contracts and relaxes, it will transmit those contraction and relaxation to suspensory ligaments, and in the process it will alter the shape of the lens. Contracts and relaxes to alter shape of the lens. So when this one happens, it will bring about focusing for objects at different distances. So this is the major role. But another role of ciliary body is to secrete aqueous humor. Secretes aqueous humor. Structure number 9. After the lens, we have a chamber, and in this chamber we have a liquid called vitreous humor. This is also a colorless, jelly-like liquid, but is found between the lens and retina. Is found in between the lens and retina. That space between the lens and retina is called posterior chamber. The roles of vitreous humor, just like aqueous humor, one of them, it gives spherical shape. Gives spherical shape to the eyeball. Gives spherical shape to the eyeball. The other one is refraction. It is involved in refraction of light. refraction of light so that they can be focusing, so that they could be focusing of light or the image onto the retina for focusing onto the retina. And remember, if you look at your diagram, vitreous humor takes the largest part of the eye for it to accomplish the very last refraction that happens. So we have mentioned three structures that will help in refraction. One is cornea, two is aqueous humor, and three is vitreous humor. But the largest bit is taken by the lens and also by the vitreous humor. Number 10 is sclera. Sclera is also called sclerotic layer. Sclerotic layer. Remember, we have looked at sclerotic layer. This one is majorly made up of non-elastic connective tissues. Made up of non-elastic connective tissues. Remember, it's the outer part. So because it's made up of, or it makes the outermost part of the eyeball, therefore the major role is giving protection to the eyeball. Gives protection to the eyeball. Eleven, the choroid layer. Choroid layer. Remember, in our diagram, we had the sclerotic layer, another layer in between, and now there was another layer made up of the retina. So we have the retina there, sclera at that point, and that middle part of the outermost layers of the eyeball is the choroid layer. But this part is characterized by being dark in color. It is dark colored. It is dark colored. And another characteristic of choroid layer is highly supplied with blood vessels, blood capillaries. Blood capillaries. So because of these two structures, then the roles of choroid layer becomes one, if it is blood capillaries supplying oxygen and nutrients to the eye. Supply oxygen and nutrients to the eye, to the eyeball. Now where there is oxygen and nutrients, we know that metabolism is going to take place, and in the process, byproducts, waste metabolic products will be released. Therefore, because of that, again, blood capillaries are meant to take away waste metabolic products. Takes away waste metabolic products. Then the other role comes with the dark color. Sometimes in the eyeball, even after those refraction, we can have some light, some stray light coming this direction. Instead of being refracted properly to fall onto that area, we can have some stray light. We can have some internal reflection, but that one does not happen. Those internal reflection do not happen because of the choroid layer. We know that one characteristic of a dark surface is to absorb light. So because of that, the light that falls on the choroid layer will not be reflected internally because it has been absorbed. So the other role in regard to darkness, second role of choroid, prevents internal reflection of light due to a stray light, due to stray lights. Number 11, we have the retina. Number 12, retina. Now this is the most important part of the eye. Even if all the parts of the eye works well, but the retina is not functioning, there'll be a problem because it's the one that contains photoreceptor cells. Contains photoreceptor cells. And because it contains photoreceptor cells, it's the one that is stimulated when light falls onto it. When light falls onto it, it will be stimulated, generates a nerve impulse, and then sends to the central nervous system for interpretation. We have two types of photoreceptors. We have those called the cones. These photoreceptors are cells. So it contains two types of cells, cones and rods. These rods are different from the cones because in each of these they have different photopigments. In the cones we have a photopigment called iodopsin. In the rods we have a photopigment called rhodopsin. The function of iodopsin is to perceive light of high intensity. Perceives light of high intensity. So how does this happen? When light falls onto the retina, when there's a lot of light, it is this type of cells, the cones, that will be stimulated. And the specific pigment that will be involved is iodopsin. So what happens? Iodopsin will degenerate into two compounds. Iodine, not compounds, into two substances, iodine and another substance called opsin, when there is high light intensity, high light. So when this iodopsin degenerates into iodine and opsin, there'll be generation of a nerve impulse. Nerve impulse is generated. That nerve impulse will travel through sensory neuron. Specific sensory neuron is called the optic nerve. It will go to the central nervous system, where interpretation will take place, and then now you can tell the kind of object you are seeing. So iodopsin for perception of high light intensity. Rhodopsin is now the opposite, perceives light of low intensity. Low light intensity. So how does this happen? It also happens in the same way when light of low intensity falls onto the retina. It is the rods that will be stimulated. It will involve rhodopsin. Rhodopsin will degenerate into two substances. We have the retinine retinine and opsin. So in the same way here, when rhodopsin degenerates into retinine and opsin, a nerve impulse is generated. A nerve impulse is formed. So that nerve impulse will go to through again optic nerve. Optic nerve. It travels, transmitted through the optic nerve to central, the CNS, central nervous system, for interpretation. Then there'll be perception of the same. But one region where we have high concentration of cones is in a region of the retina called fovea centralis. Fovea centralis. Therefore, in fovea centralis we have high concentration of cones. But rods are usually highest in concentration at the periphery of the eye. Periphery of the eye. Rods are in high concentration at the periphery of the eye. That's why at night, if you want to look for anything in darkness, most of the time you're not going to look at that object straight, but from the side of your eyes. Periphery means on the sidelines. So this is how the retina is involved in perception. So from this we can easily say that retina is the major site for perception of light, because it is the one that is containing photoreceptor cells, cones, called cones and rods. And we have seen how each one of them is involved in generation of perceptive impulses. And then we have other structures. The blind spot. Blind spot. Now there was a region on our eyeball, that point where blood vessels and nerves, where blood vessels and nerves join the eyeball. So that point is called blind spot. From the word blind, it means if you are going to have light reaching at that point, an object being focused at that point, it is not going to be perceived. Why? Because it lacks photoreceptor cells. It lacks photoreceptor cells. So because it lacks photoreceptor cells, any light falling onto this point is not going to be seen. It's also called the yellow spot. Yellow spot. Umeniambia. Also called the yellow spot. And then the very last is fovea centralis. Fovea centralis. Now, fovea centralis is a point on the retina. Remember that point. Fovea centralis is a point on the retina where there is high concentration of cones. So any image falling at that point is going to be perceived because of the cones, and is meant for perception of light. So those are the major parts of the eye. Now we can recap the same using some few questions here. One of the question is name three layers of the wall of the eye from outside. Name 3 layers of the walls of the eye from outside. We can go together. We say the outermost part of the eyeball is called the sclera. Next, the middle part that is dark in color with a lot of blood vessels, choroid layer. And another layer is the retina. Question number 2. Which parts of the eye helps to focus light onto the retina? Which part of the eye helps to focus light onto the retina? I think it was clear. When light falls on the eyeball, the first part that's involved is the cornea. For refraction, it will go through a liquid matrix called the aqueous humor. For refraction again, and then we have the lens again for refraction, and then more refraction by vitreous humor. By vitreous humor. I measure. And then the last question. Name the light sensitive. Sensitive cells found in the retina. We have said those light sensitive cells are called photoreceptor cells. There are 2 types. We said we have cones and rods. Naniambia kama inaisha. Mabaki 3. If we wake up in the middle of the night and switch on your light and then off, describe what happens in the eye. Describe what happens in the eye when light is switched on and off. When light is switched on and off. Now we said there's a structure in the eye that's involved in regulating amount of light that reaches the retina. That was the iris. Iris has 2 muscles, radial and circular muscles. Radial, circular muscles. But of course with other structure called the pupil. So the following will happen in the 2 situations. Light on. Light on. Radial muscles relax. Circular muscles contract. The size of the pupil decreases. When the size of the pupil decreases, less amount of light. So we have said radial muscles is going to relax. Circular muscles contract. Radial muscles relax, circular muscles contract. The size of the pupil decreases. So when it decreases, the high light in that room, there will be less light reaching the retina. Light off. When lights are off, for one to perceive, we need more light into the eye. Opposite will happen for each of those muscles. Radial muscles are going to contract. Circular muscle is going to relax. When circular muscles relaxes, pupil will increase. Pupil size increases. When pupil size increases, some lights or a lot of light will be allowed into the eye. There's no light at that time, but there will be accumulation of more light so that they can get into the eye for perception of the object. Thank you. We have more questions. You can always get them from the library and from the internet to help you understand even more how the eye works. Thank you.